Good evening, everyone. Welcome to the Snyder Institute for Chronic Diseases Mini Medical School Lecture Series, hosted in partnership with the Calgary Public Library. We're pleased to host a compelling presentation and discussion this evening on the topic of the war waged within, Pacman and the fight against autoimmunity. My name is Hannah Riley, and I will be the program host for this evening. Before we get started, I'd like to go over a few housekeeping items. There will be two parts to tonight's program. First, each of our presenters will speak for approximately 20 minutes, and then we'll open the floor to audience questions. We will take a 10-minute coffee break between each speaker, and refreshments will be provided to those of you here in person. You are welcome to ask your questions at any time during the presentations using Slido. To access Slido, you can scan the QR code or type www.slido.com into your browser and manually enter the event code SNYDER-MMS into the participant field to post your question. Questions submitted to Slido will be viewable to all audience members, and participants can upvote the questions they're most likely to see asked. Now onto more specific housekeeping items. For those of you who have joined online, if you lose your internet connection and drop out, you can return to the session using the same Zoom link you used to join us now. If you experience any technical issues, such as you cannot hear or see the presenters, please email snyder@buxa.com, and our team will do their best to support you. For those of you here in person, we ask that you now switch your cell phone to silent mode. If you need to use the washroom, the closest facilities are off the corridor to the left when exiting the Libben Theater. In the event of an emergency, please exit through the doors nearest to you. Once out of the lecture theater, the closest entrance or exit is the north entrance to HSC. Please note that this event will be audio recorded, and the recording will be posted on the Snyder Institute website. Now, to begin our program, we would like to acknowledge the ancestral territory of the Blackfoot Confederacy, which is comprised of the Siksika Nation, the Piikani Nation, and the Kainai Nation. We also acknowledge the Treaty 7 signatories of the Tsuut'ina and Stoney Nakoda and First Nations, and most recently, the Métis Nation Region 3, and all the people who make their homes in the Treaty 7 region of Southern Alberta. This land acknowledgement reminds us of the histories that precede us, highlights our responsibilities going forward, and helps bring us together on a shared journey of truth and reconciliation. I'd now like to introduce Miss Juliana Svischuk, who will moderate tonight's session. Juliana is currently pursuing a master's degree in microbiology under Michael Parkins here at the Snyder Institute. Her work focuses on exploring the impact of cytomegalovirus on bronchial epithelial cells, particularly in individuals with cystic fibrosis. Juliana also serves as the Snyder Institute Trainee Committee's Education Co-Director and has led several successful educational programs, one of the most recent being an interactive microscopy event entitled Living Pictures, The Art of Microscopy. Now I will hand things over. Take it away, Juliana.
Good evening, everyone. Thanks so much for joining us. I want to thank you so much for your continued support of our mini medical school lecture series. I'd also like to thank our sponsors, UCalgary's Cummings School of Medicine and AstraZeneca, for their continued support of the series. And finally, of course, I want to thank Snyder Institute members and collaborating institutes for volunteering their time to present these excellent lectures. Without their support, there would be no series. So thank you to everybody. So now I'm pleased to introduce our first speaker of the evening, Dr. Antoine Dufour, who is an assistant professor in the Department of Physiology and Pharmacology at University of Calgary's Cummings School of Medicine, and he's a full member of the Snyder Institute for Chronic Diseases and the McKeg Institute for Bone and Joint Health. Now, Dr. Dufour's research focuses on molecular scissors, or proteases, which are enzymes that perform essential functions to regulate most, if not all, cellular processes. Now, proteases act in networks to amplify signals, regulate biological functions, generate cleaved proteins or cut proteins, and are molecular effectors involved in most human diseases, including autoimmune diseases, which will be one of the topics of our discussion today. So please welcome Dr. Dufour.
Great. Thank you very much for having us. I'm very happy to partner up with Dr. Choi, who will talk after me about this game of Pacman that we thought would be one of the good ways that we could used to understand these very complex diseases of autoimmunity that are very painful and challenging to treat. And at the center of the screen, you can see my lab logo, and that was developed by an artist in a collaboration named Ron Through. And is actually watching the talk tonight from New York, so we say hi. And I'll tell you a little bit about our partnership of what this came about last year and why it became the center of our lab logo. So you can see in the Dufour, the O is a little Pacman in a kind of artsy way. The A is a little ghost. And I'll tell you a little about what that means in terms of biology and autoimmunity. So I grew up, I always liked game, and I thought that would be a nice way to understand really this complex mechanism that are happening in autoimmunity. So, you know, these commercial that you see the guy sideways or the woman like this, and they're just taking a pill and then it lights up as the pill goes down. I thought it always made me laugh. And it's like, why not make a giant game of Pac-Man? And what we're trying to do in our lab is really to understand what are the rules of this human game of Pac-Man, which is an 80s arcade game, right? So it's a pretty simple game. There's 3 components. There's Pac-Man, Ghost, and little dots that the Pac-Man needs to eat. So in this game, proteases is a scientific word to depict enzymes that are molecular scissors. So they cut and they degrade other enzymes in our body. So in the game of Pac-Man, you have the Pac-Man that's walking around, this kind of labyrinth looking. And then the ghost or the protease inhibitor are chasing it. So if the ghost hits the Pac-Man, you lose a life. And then you need to put another quarter in the machine if you run out of life. And the point of the game is to eat all the dots that are within this labyrinth. Right? So let me put numbers on this game in terms of a human body, okay? So there's 473 proteases that us humans produce in our genome. They're not all active and there at the same time, but that's the maximum number of enzymes that we have that are called proteases, right? We have 156 ghosts or protease inhibitor in our body. And how many dots, how many proteins are generated from the human genome, right? So you would think there's a precise number, but there's actually an approximate number because we don't have a full understanding of how the genome can translate to become proteins that are expressed. So one number is 20,359. That's a pretty good estimate of what we have. That's a gene that becomes a protein in our body. But if you look at a cabbage has 44,000 different gene encoding proteins. I think all of us would think we're a little bit superior as cabbage. I got nothing wrong against cabbage, but I feel we are a better or more complex species than a cabbage, right? So what's going on here? What's this complexity arising from? And if you need to put a school bus together, you're going to require about 21,000 different bits and pieces and parts, right? Well, of course, as human, we're much more complex than a school bus. So what is this complexity arises that makes us humans? So the answer is really the chemical modification. The scientific word is post-translational modifications, but they're really kind of chemical enzymatic modification. Now this number quickly becomes over 1 million different bioactive proteins. What's fascinating and what Dr. Choi and myself are going to talk about today is how much these impact human health and diseases. So we know of roughly 300 different chemical modification in humans, okay? There's molecular scissor proteolysis. That's the one I will talk about today. And maybe some of you have seen the Nobel Prize, Caroline Bertuzzi at Stanford and her fellow winners for Nobel Prize won for studying how important sugars or glycosylation impact cell and function. So those are 2 classic examples of how key these modifications are to understanding health. And often in diseases, there's dysregulation or changes in these processes that becomes problematic. That's what we're trying to understand in this giant game of Pac-Man. So all up to now, I started my program about 5 years ago. I was like 473 Pac-Man. That's not too bad to study. And maybe some of you have seen at the Snyder Institute, we have a world-recognized sets of scientists and team here studying what microbes, what viruses, what fungi do. And that's a major impact in our understanding. Bacteria, virus, and fungi, in fact, have equal number of cells or even more than we have human cells. So us as human, we're kind of less human and more bacteria, viruses, and fungi in terms of number. And that means there's a lot of non-human proteases. So that's good news for me because I'll be stuck for a while, and I'll have a job for a long time here, right? But it's fun to study, but it's also very challenging to incorporate all these together. So a little question here: if you look in a database that is vastly, incomplete, how many proteases do you think are known to exist in nature? Not all of them interact with humans, but many do, right? So the answer is over 1.1 million, right? So that's a lot of job for people in our field to understand what these things do. So why proteases? Why these scissors? Why am I so excited to work on these types of enzymes? Well, it's pretty simple. Every single protein that we have in our body will encounter molecular scissors at some point, whether it's a signal peptide, propeptide activation, processing, so things get cleaved and then it changes their function, like a receptor, like a switch that turns on, or degradation. Sometimes you need to get rid of stuff. All of those are regulated by proteases. In fact, there's nothing that is alive that can be alive without a protease. So a friend of mine in San Diego actually calculated what is the minimal amount for something to be alive and how many proteases there are. The answer is 2. So that's the very minimum you can have in a very basic organism. So proteases are needed for life. Approximately 8% of all the drugs are protease inhibitor linked to proteases or proteases themselves that are used for therapeutics. So there's a lot of interest from pharmaceutical companies as well. So some of the famous example of that is to treat virus infection, cardiovascular disorders, blood coagulations, and there's many other diseases as well. Recently, we've heard about COVID, and Paxlovid is a protease inhibitor as well. So that was the only treatment for SARS-CoV-2 or COVID, right? So there a lot of interest from pharma to develop new drugs and better characterize what proteases do in diseases. I'll give you 3 examples of cascades and proteolysis that happens in our body. The first one is coagulation. So when you cut yourself, you're going to bleed, and then of course you're trying to stop the bleeding and form a fibrin clot to stop the bleeding. So this cascade is a series of proteases, of Pacmans that cut one another to create this effect. And of course, if you have problem in your coagulation, you may be hemophiliac, and that's very important to control that, right? The second example, and again, Dr. Choi will talk and expand more on what the complement system is, but it's an important immune regulated system. Believe it or not, this is a simplified version of the cascade. It's quite complex, and we truly don't understand all the connections. So you can quickly get dizzy in trying to follow the arrows here. But this is a very important in our immune response. And a lot of those proteins are proteases. They're Pacman cleaving, impacting one another. The third example is how a cell dies, cell death. The scientific word is apoptosis for that. And caspases are some of the main proteases that cut one another and tell the cell to die. And if you don't have this process, you may develop a tumor, which is of course a problem. So apoptosis cascade has been heavily studied in cancer, but it's also implicated in several other diseases. So this is a very simplistic way to look at the protein. And when the protease, when the Pacman comes and cuts this bond, it creates a separation of the peptide bonds, and this generates new chemistry. So what we do in our lab is we can react with these open chemistries, and we can basically find needles in haystack by using chemical advantages to study that. And in every disease, there will be changes that we can follow and track. So what I want to cover today is about protease and Pacman in human disease. Can we use them as biomarkers or disease indicators? So I'll give you 3 examples of how this have shaped the history of science and what is sometimes being used in the clinic. So a biomarker could be defined as a biological molecule or protein found in blood, other bodily fluids or tissue, and that could be a sign of a normal or abnormal process. It can be associated with the disease state as well. So the first example of this biomarker is called the prostate-specific antigen, or PSA. So maybe some of you have either done a test or seen these results. So PSA is a protease. It's a Pacman. It's called kallikrein-3, but often we use PSA in the tests and the stuff that we do. So this was, in fact, the first cancer biomarker approved by the Food and Drug Administration. So the concept is actually pretty simple. You have an elevation of PSA or kallikrein-3, and you're likely to have prostate cancer, right? The concept is pretty simple, yet it's not really how it works in the clinic, right? Things are more complicated than that. 75% of men with high PSA maybe don't have cancer, and 14% of men with prostate cancer may not have high PSA, right? So it's an indicator. Is it a great indicator? Well, let's call that in a gray zone. It's arguable. But this is actually a very good example of how, it shaped really the history of biomarker discovery. And it happened to be a protease. So of course I was interested by that. Proteases are not only biomarkers of disease indicators, there's active enzymes involved in cancer and in other inflammatory disease. So I'll give you one example here. So here we have breast cancer cells, and we can color different proteins with different dyes. And here we can see in blue the nucleus of the cells, and in red and in green, we've colored different proteins we're interested in. This is without a drug treatment. And if we have an inhibitor for a protease, now you can see there's change in morphology. The cells are not shaped the same. The colors ratios start to change. These cells are more clustered. They're not as migratory and invasive, right? So we can impact the function of cancer cells by interfering with proteases. Now, this is where it gets interesting and tricky for us basic scientists when we study these processes. Is some of these Pacmans are good, some are bad. We don't know when and why. And we need to understand them individually to see, is it a body response to protect against cancer? Or it's the cancer cell that uses these enzymes, uses proteases to make the disease worse. The second example I want to talk about is C-reactive protein, or CRP. That's another markers that you may have seen in some tests that are being done in the clinic. CRP is made of 5 subunits. Often it would be elevated after 24 hours, usually of pretty significant inflammatory or infection process. It starts to rise in about 4 to 6 hours. So again, the concept is pretty simple. You have inflammation or infection. That's a sign that there's something wrong. There's something bad going on. However, it's not perfect either, right? When you get to the site of infection, there's often proteases from either bacteria or the host that changes the function of CRP. It creates monomeric CRP. We truly don't understand what's the difference between native and single CRP. What's the true function of that? We're not 100% sure on it, but although it's been studied a lot, we know it's involved in the upregulation of infection or inflammation. But again, it's not perfect. A CRP test, it won't tell you where the inflammation is located or what's causing it, right? So you do need additional tests to pinpoint what's the cause and location of the inflammation. So again, it's an indicator, but it's far from a perfect markers, and not definitely a precise marker of what's going on in the patient. The third example are the complement proteins. They're proteases. Many of them are proteases themselves. And one example of that is a complement test can be used to diagnose or monitor systemic lupus erythematosus, or lupus for short, that's often referred to. So if you have a low level of C3 and C4, that could be an indication that you have lupus. It could be an implication of kidney inflammation, which is definitely a challenging feature of the disease to treat. Again, I'm bringing back the very complex map here. I've circled in red the C3 and C4. So those are proteases. They're cut in multiple ways, and depending where they're cut, that changes what their functions are. So one of the functions I'm really interested to study is how our immune cells react and start eating bacteria, and they get the signal from the complement to say, well, I should eat that, I should eat more of that I don't need to eat. So this is a picture of a macrophage. It's a white blood cells. This is actually a picture I've taken. This is a mouse macrophage. So I've taken from a mouse and imaged it. I always like that picture. It reminds me of a sunny side up egg, when you're cooking this, but you can see there's little legs, there's little tentacles on this cell. And these cells are extremely, extremely reactive, and that's how our immune system, at least part of it, works. So these cells are very reactive to how complements are cut. Depending on how the complement system's cut, they'll go and eat it. So if you add in pink, you can see fluorescent beads, and we can cover them with different complement and cut the complement in different ways. And we can see how many beads have been eaten by this white blood cell. So you can see here a top view and a side view. And in fact, when you zoom in on these processes, it's really impressive. It looks like a crab or a lobster grabbing these things, and they go around it and they grab it like a hand, and then they eat it up. This process is called phagocytosis, and that's how we recognize antigen. In this case, in this experiment, what I had done here is I cut the complement a certain way that makes these cells very hungry and they just want to eat it. So that's what I was testing in this experiment. But this image is pretty cool. I've taken it right underneath, and then it just grabbed these things. And then you can monitor in real time and you see them eating them. So that leads me to the art and science collaboration. So I've given this talk in an artist called Ron Through. He's watching us tonight. So from New York. So we say hi to Ron. You can check his website and his Instagram. The artwork that's outside is actually done by Ron. And this came from an effort from Valerie and Juliana, who are a graduate student here. And they were always interested to use art to better describe, better understand science. And there's a lot of interesting art. So when I talked to Ron, I've given a similar talk in New York, and I had the chance to meet him. And I told him about, I need a new lab logo, and I'd like for you to do something about our research. And he asked me, well, I'm not a scientist. Like, what can I do? I was like, well, you know what, this Pacman game with this ghost, just go artistic on it and come up with fun design. And Ron was fantastic. He came up with a very interesting design that is his point of view of what I've been telling you today about. So this is some example here. You can see kind of a pop art thing of a Pacman kind of eating the ghost with a sign gulp. This one is about the social media of how do Pacman and ghosts talk and interact with one another. There's a huge social network of signaling that's going on all the time in our body. And that's what I think Ron was trying to depict here. Ron had written a great book about Roy Lichtenstein, who's who invited pop art, who was actually a professor in New York at the time. So inspired by that, he created something with dots that resemble a little bit of Roy Lichtenstein's paintings. This is the one that you may recognize from our lab logo. So we've taken the background and we put it on a white background. This is the Pacman and the ghost that ended up in our logo. So that was the winner when our lab voted on what logo to pick. So we've been doing research in our team for about 5 years in Calgary, and there's a few lessons that we've learned so far about this human game of Pacman. The first one is in cancer and inflammatory disease and autoimmune disease, we tend to have more Pacman showing up and less ghosts. I don't know why that's the case, but it seems there's a need or there's a change and there's more Pacman than ghosts present. Now, like I said, the challenge is to understand which one of the good Pacmans, which one of the bad Pacman, and we need to characterize these proteins in a much more careful way. They're also dependent on what disease you're looking at. If you're looking at an arthritis situation or a cancer situation, those are different scenarios, and you're likely going to have different Pacmans showing up. You know, very impactful work done at the Snyder Institute shows that the viral, the fungal, and the bacterial proteases are absolutely important for our health and often are dysregulated in disease. So there's not only the yellow Pacman, there's other types of Pacman that are involved in our disease, in our homeostasis, in our normal health. Now, the substrate, the dots, are not like in a classic game of Pacman that are nicely distributed. They're in clusters. These dots are colored in different ways. You know, like Caroline Bertuzzi with her Nobel Prize this year showed how important sugars are and how much they impact our health. Well, sugars also changes the way proteases can eat the dots. Sometimes it makes it easier to eat, and sometimes it makes it harder to eat. And there's a lot of those modifications. So dots are not evenly spaced. They are in clusters and they're very messy. To understand the dots is a nice window to understand disease as well. So this is a very important point that a lot of labs around the world are studying as well. And lastly, it's really, we truly have an incomplete understanding. Out of 473, we maybe understand 150 of those Pacman. There's tremendous amount of work left for us to understand and characterize what these Pacmans do. So we do truly have a incomplete understanding. There's a lot of gaps of knowledge in this game. So there's a lot of bits and patches that we just can't see, that we need to study and better characterize so we can either understand a disease or maybe find new ways to target a disease with a drug. And I'll end on this picture that I think represents very nicely what we do in our team, in our lab. This is a piece by Shikio Fukuda, who's a Japanese artist. Unfortunately, he's passed now, but I'm a big fan of his art. And he put a bunch of garbage that he collected in Tokyo on a bench. So this is not artistic at all. There's nothing artsy about that. And that's really what we feel as scientists when we're collecting data. A lot of it just don't make sense yet. You need to take a step back and look at the entire picture. And all of a sudden, there's a very different picture emerging, right? So what's important is not the individual parts that you're studying, is really how do those parts interact with one another? And now there's a very different picture that emerges in these interactions. That's where we need to get at. That's what we need to understand in these diseases. So I'd like to thank the team in my lab that are collecting and are working on these data. Also the funding that we have, so we can play around in the lab and run some fun experiments. So thank you very much, and I'll be happy to answer a question in the later part after Dr. Choi gives her talk. Thank you very much.
Thank you so much, Dr. Dufour, for that wealth of information and wonderful presentation. Thank you. So we're now going to take a short coffee break. So please feel free to step outside for about 10 minutes. And yeah, we'll see you back at, I'd say, 7:10. Yeah, see you soon. Thanks. A moment for everyone to get settled. Thanks so much for rejoining us. I hope you had a chance to grab a snack and a drink. So now I'd like to introduce our next speaker, Dr. May Choi, who is a rheumatologist, clinician scientist, and assistant professor at UCalgary's Cummings School of Medicine. She's also an associate member of the Snyder Institute for Chronic Diseases and a full member of the McKeag Institute for Bone and Joint Health. As an early career investigator, her research is focused on biomarker discovery and validation for the prediction of clinical outcomes in autoimmune rheumatic diseases and the prevention of autoimmune disease development and disease-related complications. So please join me in welcoming Dr. Choi.
So good evening, everyone. Is my mic okay? Perfect. Louder. Okay. How is that? A little bit better? Okay. I'll try my best. So good evening again. Thank you so much for inviting us to give a talk tonight. I'm really excited to be talking a bit more about autoimmunity and the very exciting research that we're involved in. And just before we start, I just want to get a sense of how many people in the audience have actually played Pacman before. Wow. Okay. Okay. Did you see that, Tony? Well, I was just saying that neither Tony or I have actually played Pacman before. So I hope I don't fail tonight. I did do some research though, so hopefully I'm actually making sense when I talk about Pacman. So the title of our talk is The War Waged Within, and I'm going to continue this analogy of using Pacman to explain autoimmunity, and that I think that this is a great way of showing how my research and Tony's research can combine to find something really interesting and show you some of the cool results that we've been working on today. And so this is sequel to Tony's talk. So the mission objectives are: one, to talk about the history of the war within. So really, what is autoimmunity? Two, identify the enemy. So what is the role of biomarkers in autoimmune diseases? And then thirdly, we're going to be talking about a new weapon who's going to help us analyze biomarkers, and that is artificial intelligence. So let's talk about this war. So normally, our immune system is exposed to a large variety of foreign substances we call antigens. Bacteria, viruses, including SARS-CoV-2, which we know very well, parasites, fungi, cancer cells, toxins, and drugs. And we are constantly being bombarded by these foreign substances. So in a healthy immune system, we're able to defend the body by attacking these foreign substances. And we can enlist the help of things like proteases, like Dr. Dufour's work, nucleases, and our body produces antibodies to tell these proteases and other proteins where to go. So here the antibodies have attached to the viruses or bacteria, and then the proteases go, hey, I see a foreign substance there. Let's go attack it. Mom. Delicious. Success. However, when we are unable to discriminate between self and non-self, the immune system goes array and starts attacking our individual own cells and tissues. So the autoantibodies that are highlighted here in yellow, those are being produced and then tagging on to our own cells and tissues. So here, it's tagging on to the heart, the kidneys, and the GI system, or the gastrointestinal system. So that's a problem, because then we're going to start attacking ourselves. And so autoantibodies can cause tissue injury and disease damage by inducing inflammation at the site. So here I have some red proteases. And Dr. Dufour talked about the complement system. So remember that very complicated figure that he had where he circled C3 and C4. So some of those enzymes in that cascade get super activated and then cause a lot of inflammation in various parts of the body, depending on where the autoantibodies are flagging. So a bit of inflammation is fine. Like, we need that to help destroy these bad bacteria and viruses. However, in autoimmune disease, this inflammation gets out of control, and we can get uncontrolled inflammation affecting any part of the body, and we need medications to help control that. Autoantibodies flag cells to be ingested and destroyed, and can also interfere with normal cellular functions. So essentially, out of order. I'm doing okay so far, right? That kind of makes sense? Okay, great. So millions of Canadians are affected by autoimmune diseases, and there is evidence to suggest that the rates are on the rise. You probably saw on a slide when you came in that they said there are today probably 80 to 100 types of autoimmune diseases that have been identified. And for the types of autoimmune diseases that I see as rheumatologist, so systemic autoimmune rheumatic diseases, it's about 2 to 5 cases per 1,000 Canadians. So let's talk about an example of a disease that I see on a regular basis. So systemic lupus erythematosus, or lupus short. It's a chronic systemic autoimmune rheumatic disease that can affect virtually any organ in the body. So remember in that Pacman, the maze, any part of that could be affected in a patient who has lupus. Another name for this disease is called the disease with 1,000 faces, because patients can present with a variety of symptoms, and not one patient looks the same. Some patients might come to my clinic with just skin and joint problems, and they're called lupus. Someone else might come in and they just have kidney disease. They're also called lupus. Somebody might come in with neurological problems with lupus in their brain. They're also called lupus. So it's very confusing. They can present with lots of different types of symptoms. The other thing that's concerning is it predominantly affects young women during their reproductive years. These are often patients who are trying to build their career and build a family. We have to take into consideration the types of medications we give them, because many of them can affect the fetus or affect their fertility down the road. So this is a major concern, and this is true for many of our other types of autoimmune rheumatic diseases. It is also more common and severe in non-white race ethnicities, Indigenous, Asians, Hispanics, and Blacks. So underlying a huge public health concern. So the prevalence of lupus is increasing in Canada. As you can see, the orange dots, especially for females, is going up. And lupus is a major cause of death in young women and is often underestimated. And here are some stats I've listed here. So what are our patients dying of? So in the past, it was definitely due to active disease, and now we have some medication to help manage that. A lot of our patients now are dying from complications of the disease and treatments, including infections and cardiovascular disease, such as MIs and strokes, or myocardial infarction, heart attack. And even though it's 2023, many of our patients still experience severe symptoms and or complications from the disease itself or its treatment. And currently, like many of our other diseases, there is no cure for lupus. However, recently, new treatments have been approved, offering new hope for our lupus patients. So this is a timeline that shows you at the beginning in the 1940s and 50s, there are lots of drugs being approved, and then some drugs being used off-label. But for a long period of time, lots of drugs were failing clinical trials, and not until recently in 2021, we were getting more treatment options for our patients, but still not a ton, and a lot of our patients are still struggling. Part of the problem is because we don't really know what causes lupus. So the accepted thought is that there are some genes that put you at risk for developing the disease. And then along the way, these patients who have these susceptible genes get exposed to environmental factors that lead to immune dysregulation. And so autoantibodies, like I showed you in an earlier slide, they're being produced, inflammation starts to happen, and then you start to get damage in your organs and tissues, and that leads to symptoms, and that is when patients get diagnosed. So this is where we're kind of seeing the patients in clinic. What we really want to try to do, though, is try to capture the patients before they develop damage. So there's this phase in between where patients don't have symptoms, but there are things in the blood that you can actually detect. Inflammation, autoantibodies. We want to try to catch these patients in this period before they get damaged. Let's talk briefly about the environmental factors because it's a very important passion of mine. And I've done quite a bit of a few studies on looking at which environmental factors increase your risk of getting lupus. So here are some of the ones listed here that have a lot of science behind them, so like smoking, BC, infectious agents, medications, vitamin D deficiency. And recently I published a study with my collaborators down in Boston, where we showed that nearly half of the lupus risk might be reduced if you adhere to modifiable healthy lifestyle behaviors. So moderate alcohol drinking, healthy weight, non-smoker, good diet, and exercise. So even though there is a very strong genetic risk that's associated with lupus, you can decrease your risk by 50% just by adhering to these healthy lifestyle behaviors. Recently, this past September, we also published a study that showed that having good sleep hygiene may also be protective. So get that 7 hours a night, guys. Okay, so this is going back to how we're too late when we see our patients in clinic. 30% of patients with lupus already have kidney disease. 50% of scleroderma patients, another type of autoimmune disease, already have severe lung disease within 3 months diagnosis. 30% of patients with rheumatoid arthritis have joint damage that's irreversible. So really, we're kind of recognizing patients too late, very advanced stage already. Going back to lupus, average time to diagnosis from onset of symptoms is about 4 years or 10 consultations with a doctor. This is a worse problem for children, males, and elderly onset because it's not because this is a disease mostly of, like I said, young women. Leads to unnecessary testing, inappropriate treatments, patient uncertainty and anxiety, again, leading to multiple medical referrals. And at the end of the day, increased healthcare costs. And here again are some of the sequelae of delayed diagnosis, in addition to the healthcare costs I mentioned. So we're recognizing patients here, but we really want to move it to the preclinical lupus. And that's when we should be starting to counsel patients, hey, you're at risk of getting lupus. You should start to modify some of these unhealthy lifestyle behaviors. This is a pyramid that essentially shows it's the same message. We really want to be getting patients, when they don't have symptoms, we know that they're at risk. We want to assess their risk, detect their disease earlier, allow for earlier intervention before they become diagnosed with clinical lupus when they have symptoms. So how can we do that? Well, we know that there are biomarkers that are detectable in the blood. We know that we can assess somebody's genetic risks by looking at their genes. We can collect their blood and check for autoantibodies. We can check for proteins and levels of inflammation. All of that can be tested. This is a very important study that was published in New England Journal of Medicine that shows you that autoantibodies can be detected in the blood years before a patient is diagnosed or even before they have their first symptom of lupus. And that's same with some of the more specific markers of inflammation. So yeah, patients before they complain with a rash and joint pain, 5 years before they come in and you collected their blood sum, there's probably already evidence that they're going to develop lupus. And then this is another paper where we did a more broader search of the literature. We showed that autoantibodies have been reported in patients over 9 years prior to disease onset, so almost a decade before they even have the disease. So very complicated because, as Dr. Dufour mentioned, so many different proteases and biomarkers to consider, add in genetics, add in age, sex, ethnicity, race, so much data. How are we going to make sense of all of this information? Look how confused poor Pacman is. So this is a typical day at work. We just get lots and lots of data generated in the lab and generated from the clinic. We're collecting clinical data on patients. So how do we take all these numbers and make sense of it? So now we have this new powerful weapon called artificial intelligence. It's a new approach to interpret and apply rich big data autoantibody data sets to clinical care. And these data sets are just getting bigger. Patients are still coming into the clinic, giving us more information. We're still collecting their blood. And every single visit, we're just accumulating more numbers that we need to crunch. So information overload. Artificial intelligence and machine learning can help us identify meaningful patterns in the data. And these data can interact with each other too. So it's able to look at potential interactions between all of the different data points. All of that would be very challenging if we use traditional statistical methods. So this is an example of a study where we took 805 lupus patients who had annual visits from the day that they were diagnosed, collected their blood samples, and tested them on with a panel of autoantibody tests. And so over 30,000 data points that we had to consider, over 700 different variables every single visit. So it's very complicated, but with artificial intelligence, we were able to condense that into the following results. We identified that there were 4 different lupus subtypes based on their autoantibody profile. And within these subtypes, each of these subtypes was associated with different disease severity, different types of symptoms, treatments, and some of them were actually had better survival than the other cluster groups. So this is just a list of all of the autoantibodies that we had tested. So you see lots and lots and lots of tests. And then we'd use artificial intelligence to basically go through that information and identified 4 different clusters. So here is this cluster graph that shows you 4 different colors, each representing a different group of lupus patients. Each dot is a patient. And essentially, like I had mentioned, each of these clusters was associated with different disease severity, types of organ involvement or symptoms, treatment levels, and also predictive of their survival. So we can essentially take that and apply it to every single autoimmune rheumatic disease. You don't need to just measure the proteins in the blood. You can do that with tissue, urine, joint fluid, and you can look for all types of autoimmune or all types of biomarkers and plug it into machine learning to identify which biomarkers are the most important. And that can also tell you a little bit more about the disease pathogenesis. So what is the underlying problem here? And so just to end this off with what my research program is doing, we're essentially trying to make this process more efficient. So we have a lab here that collects blood samples from various research cohorts around the world. So when the researchers see the patients in the clinic, they collect the clinical data. The patient goes to the lab, gives their blood sample. That blood sample gets sent here to Calgary, and we put it in our freezer. And when we're ready to test the samples, we run them through a panel of autoantibody tests. And then we use machine learning right away to try to understand that data. So that makes it very, very efficient. And so these are just some of the research collaborators that are working with us and the diseases that we're collecting in our biobank, just down the hall upstairs there. So going back to the Pacman, to establish peace with our immune system, I've hopefully demonstrated to you that an earlier and accurate diagnosis of autoimmune diseases is needed. We're clearly too late when we see our patients in clinic. They already have organ damage. Implement risk assessment and preventative strategies at the preclinical phases of disease to prevent it from even progressing to that stage. And then we talked about how important biomarkers are and how artificial intelligence may play a role in interpreting those results to help us eventually identify a cure. So thank you.
Thank you so much, Dr. Dufour and Dr. Choi, for the fascinating information you shared. So we're now going to open the floor for questions and answers. We'll do our best to alternate between questions from our virtual and live audience members. So as a reminder, questions can be submitted virtually through Slido by scanning the QR code here, or by opening www.slido.com and entering the event code Snyder MMS into the participant field. Now for our live audience, please raise your hand if you have any questions, and someone will provide you with a microphone. That'd be Hannah. And we'll do our best to answer as many questions as we can within the next 30 minutes. You want to start with the live, or do you want to start with the library? All right. All right, so we'll start with the first one. It's quite relevant. So I guess to both of you, Dr. Dufour and Dr. Choi. So there's a theory that long COVID has an inflammatory aspect to it. So, do you know what the mechanism of increasing inflammation would be?
As it is a new disease, I think there's still a lot of question marks. I think we've seen a lot of lab worldwide investigating this. So I would say it makes sense to imply inflammation, especially damaged organ in some patient as well, especially if people have been hospitalized for a long time. So inflammation, yes, there's might be fibrosis happening, or maybe both together. So I think it's probably case dependent. It's probably due to if the patient has been hospitalized or not, for how long this person has been hospitalized. The age of the patient may have an impact as well. And I think it's too new of a disease to truly, I think, understand what's the best way to treat that yet. I know there's a lot of research, not only in Canada, but all around the world, to better characterize what this concept of long COVID is. And one thing that I find personally interesting too is that what if people have been infected with COVID 2 or 3 times? Does that changes that? Are you better immune? Does that how does that work? And initially, we've seen these kind of horror stories of patients with very severe symptoms and things that remain, whether they lost their voice or their lungs. So yeah, I think it's. What do you think, in your opinion, too?
Yeah, I totally agree. I think we're still doing a lot of research on long COVID at this point. And if you think back to my slide about, the foreign substances and how COVID viruses can, it is a type of foreign substance that then attacks our body. And our body then, if it reacts abnormally and produces these autoantibodies and starts to attack our own cells because it makes a mistake in recognizing our own cell as a foreign substance. And then potentially that virus has triggered an autoimmune response. And there's a lot of research to see whether or not in the long term, if someone's been exposed to the COVID virus, that they will develop autoimmunity down the road. And so that's still an unanswered question. But a lot of viruses, a lot of people do think that autoimmunity is triggered by environmental factors like viruses, like COVID-19. So I think we will find out as we do more research in this area.
I just want to add one more thing in terms of the theme with the Pacman and the proteases. So SARS-CoV-2 has two new proteases that has been heavily studied by us, protease nerds. What's really interesting to me is that initially, two proteases were involved, post-protease, so ACE2 and TMPRSS2. Maybe some of you have heard of ACE2 and from the news and stuff. That was the initial entry points. There's been other Pacman implicated in that. And now with mutants and new version of COVID, there's new proteases involved, like cathepsinL, just, that is a different word. And we don't truly understand how the virus is evolving to maybe adapting to our immune response. So that's quite fascinating in terms of a protease perspective. And long COVID may be different now than it was a year ago, 2 years ago. And I don't think we have the answer for that, but there's a lot of interesting science to follow up on that for sure.
Thank you so much. Do we want to take a live question, Hannah? You one in the corner here.
My question has got to do with the organization of what you do. So there's an implication of some pretty sophisticated lab work. There's the medical research, I think that you're representing. There's the ability to apply AI to it. Could you comment a bit? How do these teams come together? What do they look like?
Yeah, I can go first.
Yeah, go first.
So going back to that slide, I don't know if you remember the wheel that I showed you. So, there is the bedside where we see the patients and the clinicians, so the doctors who see the patients. Sometimes we get approached by doctors who have really interesting questions that they want answered. And they start to collect large groups of these patients, like lupus patients, and then they can build a research cohort or a group of patients with lupus who they follow for a long time. And then they can build databases where they collect information on these patients, like how long have they had lupus for, age, sex, ethnicity, race. And so when they collect that data, they often also collect blood samples, sometimes tissue, urine, and all of that gets sent to our lab where those specimens are being stored. We actually just do blood right now, but there's opportunities to collect other types of tissues. So our lab gets approached by researchers from Canada, internationally, in the US, and they often ask us really interesting questions. They're like, oh, we're interested in this particular biomarker that we think may be helpful in helping us diagnose. Can you test that for us? And then we'll say, okay, yeah, sure. Send us some of that blood sample and we can test that particular biomarker. Or if we don't have it, we actually have the ability to develop the assay or the test to test for that protein in the blood. And then the novel aspect of our lab is that we have machine learning experts who are readily available to take the results that we get from the lab and combine it with the data that we have in the clinical side of things, and then interpret all of that data together to come up with some interesting patterns and results. So it's really a big collaboration with everyone. I think it starts with the patient, seeing the patient, coming up with interesting research questions. And then we just make sure that we're kind of available. We go to conferences to meet other researchers and see what their interesting research questions are and see how we can help them answer them. So I don't know if, Tony, there's anything else you'd like to add?
No, I think that was a great answer. One thing that is quite fascinating is sometimes the computers and AI will detect patterns that are completely new to us and we don't understand the biological meaning, yet it can be very informative for the clinical diagnosis or scientific question. And sometimes you can generate new hypotheses by observing patterns that are completely new. And the computers can be very good at doing that. Whether we understand what it means, that's another question, but it's quite interesting. So, but it's a very dynamic area of research and clinical monitoring now that we're going to see more and more of it for sure.
Yeah, and then the other thing that we didn't really touch upon is after we get the results, we present them at conferences. And so we share that with other scientists, researchers, doctors, and sometimes patients also come to share our findings and then generate even more questions that we then bring back to the lab and try to answer.
Thank you. So we'll take another question from Slido here. So one that's been upvoted a couple of times. Is it just lupus that you can detect inflammation or inflammatory markers in the blood, or are there other autoimmune diseases that can have their inflammatory markers tracked?
Oh, sure. So definitely, I just talked about lupus tonight, but almost all of our systemic autoimmune rheumatic diseases can have inflammation detected. So you could definitely mark inflammation in the markers in the blood is very helpful in the diagnosis, but also monitoring disease, because that's the underlying problem, is that the immune system has uncontrolled inflammation in the body such that you can even detect it in the blood. So often patients, when they come in, they're like, oh, my joints are really painful, or I have this rash. And they think that that's where the inflammation is, but actually that inflammation is running throughout their entire body. You know, that you can detect that in the blood that's running through their body. So not just lupus, but also myositis disease, autoimmune disease in muscles, rheumatoid arthritis in the joints, scleroderma, which we had also talked about with lungs and skin and also joint. So it's really a systemic problem, inflammation, and then we give patients immune suppressing drugs to try to calm down that inflammation.
I'll just add another quick part. So I think there's a lot of disease that can be used with these panels, biomarkers and stuff. One of the challenge, I think, is the heterogeneity. So patient may present with different symptoms, like in the case of lupus, or pain is another thing. Pain is subjective, like it's not the same for everybody. And sometimes people will have no pain, therefore they're not going to go see their doctor. And some people may have a lot of pain with minimal damage. So the, individual patient will have very different response to drugs as well. So those are important factors that one need to incorporate in the diagnosis or the treatment. So inflammatory markers are like some of the example I gave. It's indicative, but is it always indicative? The answer is often, no, it isn't. So you may need to do other tests and continue following up the patient to better characterize or follow up if the drugs are working and not, and change maybe your treatment plan or not.
Thank you. Do we want to do another live question from the audience?
Great questions, by the way.
There's been a lot in the media lately about AI and machine learning, and a lot of like fear mongering a little bit, I think, going around. How do you think that's going to affect your research and your ability to do some of these projects and to treat people?
That is a very valid point. And I think that, AI is already here. A lot of the things that we're doing, machine learning has already taken over. So I think it's just going to be inevitable that we're going to use AI more and more. And why not, if it's able to help us find a cure to a disease eventually. But yes, you do bring up a good point that we have to consider things like patient confidentiality. If you're going to put all that data into a big data set and try to come up with patterns, but more data you have, the better. So you're going to get a lot of information about people. And that's one of the major concerns with doing artificial intelligence. So I could do an entire talk just on that, but just for tonight, I think I'll just say valid point. But I think right now it's something that I think is really cool and will help us a lot in medicine.
And ChatGPT just came out, and you can that's the one that I'm teaching a lecture and students to talk about a disease they're interested in. And you can put that in ChatGPT, which is an AI program that will tell you everything you need to know in less than a minute. And the problem with that is that it uses information that's out there, but not information is accurate. Another challenge with AI is that it's as good as what you feed it, right? So there's a lot of unknowns and gap of knowledge that we do have. So this is not going to help us for that. Now, the good thing though is that the speed that it can accomplish and make connection is so advantageous that we can absolutely use that to our advantage. But it often depends on the quality of the data that you feed it, right? So I think there's a balance there. So it's not going to take over our job, but you can absolutely use that as a tool to help us make right decision either in research or in the clinic.
Thank you. We'll ask the next sort of upvoted question. I'll say upregulated question. Is AI being used in Dynalab at this time? For example, can a physician test for autoantibodies with AI to help determine the type of SLE?
Not at this time, but it would be very cool if you could eventually do that.
Thanks so much, Dr. Choi. Hannah, do we want to take another live question? Back there.
You pointed out how lupus can present itself in very different ways. And I presume that the practitioner who sees the patient is going to do different tests depending on what he sees. Do you feel confident that you can separate out the test data that comes and indicating the way that the patient is presenting from the underlying cause? Or is this likely to be something which perturbs the AI? Did I explain that?
Maybe just this last part. Can you explain that last part of your question?
Okay, I'm concerned that different patients may have different tests done because they're presenting in different ways. And yet you want the AI to come up with one result for all of these people to say that they have lupus. How can you avoid that?
Yep. So I think there's probably two parts to my answer here. I think one of it is, as you pointed out, some patients are getting a lot of different tests and not all of them are going to be useful. And so it could be a potential waste of health care resources. The patient might undergo unnecessary testing that could be painful. For example, getting a biopsy that they might not need. They might just have to wait a long time to get these results done. So I think the point of AI is try to find out exactly which tests are the best ones to get, instead of just ordering a bunch of random tests that may or may not be helpful, or ordering too many tests when you only maybe just need a few. So AI is trying to help us kind of, take out the noise and just focus on the best tests that we should be ordering.
Thank you. We'll ask another question from Slido here, and we'll kind of tandem these two because I think they can go together. So if a person has no symptoms, is there a routine blood test which could indicate a need for other testing regarding early diagnosis? And sort of on the other end of that, what are some treatments indicated for patients that are positive for early signs of autoimmune disease? Dr. Choi or Dr. Dufour?
You can go with it.
Lots of. So if a person has no symptoms, is there a routine blood test which would indicate a need for other testing? Yes, that's a good question. Usually we don't send a patient for a routine autoimmune testing if they're not presenting with any symptoms at this time. There's a test that gets ordered quite a bit. I don't know if you've heard of the anti-nuclear test. And that's used to screen for autoantibodies. But right now it's not recommended that you order it just because if you have no symptoms, because there are lots of reasons for that test to be positive. About 20% of patients who have a positive ANA might just be healthy patients. Sorry, healthy patients. They could just have a positive ANA, and 20% of ANA people who get the test are actually healthy, so falsely positive. So no, there's currently no routine blood tests. But the goal of my research is to eventually find, once we've assessed these patients who are at risk through genetic factors or environmental factors, that we could identify a blood test that could further risk stratify them to say that yes, you are at high risk of getting X disease, and so you need to be doing X, Y, and Z to lower your risk for developing disease in the future. And then the second question is, what are treatments? So right now there is, look, I can say that most people would say modifying your lifestyle would be, it's not a medical treatment, but there's been, like I showed you, stopping smoking is helpful, exercising, certain diets, the Mediterranean diet. I would always kind of give my patients that kind of information. If they come to my clinic, I haven't given them a diagnosis, but I feel like they may be at risk. I would say you're in early stage potentially, but and these are the things that you can do. But there's no magic pill that they can take that would stop them from progressing. At least we haven't discovered anything like that right now.
And I'll just add, in the routine blood test, there's many things we can look for, but we cannot look for everything for every single patient if there's no symptoms, right? Because it would cost time and money, and it's not fair for patients that do need the care, right?
Yeah.
So again, you need to know what you're looking for to find it. And currently, we're not testing for 500 different things. A routine blood test won't tell you a lot of the extra information. It'll give you kind of basic things.
Thank you so much. Do we want to take another live question from the audience? Okay, well, we don't seem to have one just now, so we'll move on to our Slido questions. I guess a bit more from the science side of things. So has there been any UFC research into potential autoimmune diseases like PMDD, endometriosis, since they've been shown to have autoimmune links?
Yeah, with the Snyder Institute for Endometriosis, we have a collaboration with Dr. Donna Slater. And yes, there's interesting autoimmune links here. That's absolutely correct. There's still a lot of characterization about this disease and how things work to better understand it. So a lot of our work on the basic science side is to understand these mechanisms that are driving or impacting, which is really a fascinating area of research. There's just a lot of unknowns still, right? So we're building that knowledge base by doing investigation, by looking at patients' sample and describing what is going on, the severity of symptoms, or how drugs work and what options we have. So absolutely, and there's a lot of activity with these diseases at the Snyder Institute right now.
Mm-hmm.
Thank you, Dr. Dufour. Are there any questions from the audience at this time? All right, well, we can move on. A really interesting question. I like this one a lot. With the AI or machine learning sort of approaches that you've discussed today, are there any collaborations with the computer science department, or really any sort of, I imagine, kind of data science perspectives to expand your medical databases or understand that technology?
Yeah, absolutely. There's absolutely a lot. I mean, I think in every university, there's been a lot of collaboration with our computer science department. I just want to point out one of the challenge so I think many people use these kind of approaches to better characterize their data and understand the patterns. Just to give you an idea, with effective machine learning, sometimes you may need 1,000 or 10,000 patients to get a good predictive pattern. But what if you're working with a rare genetic disease that you may only have 5 patients in Calgary? That becomes very challenging to use these tools because you're just missing data points. And then it becomes very challenging to create predictive patterns that are helpful. So I think it depends on the disease you're working on, and population health. And for example, one example would be COVID. A lot of us have been vaccinated, and there's a lot of people that have been impacted by that. So these kind of disease would be a great strategy. It is being used not only by companies but also universities to better identify patterns. So this is absolutely an active area of research all across Canada and the world.
All right, we have questions in the audience. How about, can we get the microphone up so everyone online? I think we need the microphone to have the folks on Zoom be able to hear. Sorry, thank you.
Thanks a lot. Hey, the average time to diagnosis for ankylosing spondylitis is like 8 or 9 or 10 years or something like that. I wonder if there's any inflammatory markers that you can see beyond, I guess, the HLA-B27 gene test, but are there things that you can see for inflammatory markers for ankylosing spondylitis and those kind of diseases, rheumatoid type diseases? It's a two-part question. I was going to follow up with something else too.
No, I think, so ankylosing spondylitis is part of a group of what we call seronegative.
Mhm.
And so seronegative means, no biomarkers. In fact, but you're right, HLA-B27 is one of the potential biomarkers that they are, at risk of getting ankylosing spondylitis. But it is, your point is very valid in that there are a lot of autoimmune diseases that we're still searching for good biomarkers. There's CRP and ESR. You're probably familiar with the terms that are markers of inflammation that can be detected in patients with ankylosing spondylitis. So we don't have anything more specific at this time, but really great area of research that we should be looking more into and trying to identify more specific biomarkers just for diagnosis and also for prognosis of disease.
Yeah, no, thanks. And as a follow-up, do you guys kind of look at the treatment side of things? Like, so I mean, for AS, there's the IL-17 inhibitors, or like for autoimmune hepatitis, there's the thiopurine. Do you guys look at that side, or it's more on the
Both.
oh,
Yeah, so we're also looking for markers to help us monitor disease. So another very important part of our research.
And one aspect following to that, so one of the challenge, and what I think is interesting, is some patients respond well to some treatment and others don't, and we don't really know why that is. So yeah, sometimes there's option number 2 or option number 3, but what if you had option number 3 and you still have pain and there's still problems? That's something we're very interested in characterizing. So some disease that affect more people is easier because you can recruit more patients. Some diseases are a little bit more rare, and then you don't have a huge sample set. So you need to collaborate with teams in the US or have larger biobanks of samples so you can, explore a little bit further out. But it's a very interesting area of research of why some people respond well and why the symptoms can be so different. Especially lupus is a great example of that. You have really a diverse sets of symptoms and markers that you can follow.
well, thanks for your answers. Thanks for your work.
Thanks for your question. So I think we have time for about one more, and we'll choose. Let's do. I feel like the third one might be kind of interesting, right? Which one do you think? Yeah. Well, we'll ask, does fibrinogen or amyloid play a part in organ damage or an autoimmune disease?
So over time, if you have chronic inflammation, you can start to get amyloid deposits and fibrosis and lead to organ damage that way. So yes, it can. And then fibrinogen, I don't know if you wanted to answer because you mentioned fibrinogen in your talk.
Yeah, so those are some example. It's often much more complex in terms of what you can do. So when you have a lot of inflammation, things can turn out to be fibrotic, and that's very challenging. We have a lot of great drugs to fight inflammation. Fibrosis is kind of the other side of the coin, and we don't have a lot of great drugs to reverse or treat that, almost none. So for a fact, pharmaceutical companies are really investing a lot right now to better understand how we can either inhibit or reverse or interfere with that. What I found really interesting is it's not a one or the other. There's a lot of overlap. If you have a lot of inflammation, you may have these fibroses that are challenging to treat with collagens, for example, deposition, and you have issues with organs, but there's still inflammation at the same time. They kind of work in tandem or they compete. And that's something we don't truly understand of when is this switch happens, why is it inflammatory and all of a sudden it becomes fibrotic? And is there a middle ground of this? And I don't think we have a true understanding of how this works, because the problem is when you have organ damage that becomes irreversible because it becomes fibrotic. Then what do you do, right? It becomes I think a different treatment altogether. So it's a very active area of research by pharma right now, because we don't have great drugs to affect fibrosis, which are involved with fibrinogen, and many other molecules as well.
Thank you. Are there any more questions from the audience? Oh, there's one. Hannah, be our last question of the day.
Is it possible to be diagnosed with an autoimmune illness, such as lupus, with a negative ANA test?
That is a very good question. And I think you heard my mentor, Dr. Fritzer, over there, who just said it's a good question, because that is basically like my thesis. Yeah. And I could definitely give you probably like a 5-hour talk on it, but essentially, you can definitely have what we call an ANA negative lupus. And there are lots of factors that can cause you to have a negative ANA. It can depend on your assay. It could depend on the type of treatment that you're on. Maybe you're on steroids and that's suppressing your immune system, so your ANA test is negative. It could depend on which part of the disease course you're in. In one of my thesis project, I basically showed that at the beginning of disease, almost all of the patients were ANA positive, like 98%. But over time, that ANA test, the concentration level, you can say, slowly declines and more patients become negative. And some patients who were tested on one assay were frequently more negative than if they tested on another assay. So lots of different factors, but it's rare, but it can happen. Yeah. And lots of these factors need to be taken into account. Thanks for asking that question.
Thank you so much to our presenters, Dr. Dufour and Dr. Choi. Let's give them one more round of applause. Thanks also to the Snyder Institute for Chronic Diseases, the Calgary Public Library, Buxa, AstraZeneca, and the Cummings School of Medicine, and to each of you in the audience for attending tonight's Mini Med School seminar. So this lecture was the fourth in our 8 Mini Medical School lecture series. So we hope that you thoroughly enjoyed it. And we invite you to join us on March 13th for our next seminar that'll be on viruses that cause cancer. So thanks so much for joining us and have a great evening and a safe trip home.
Thank you. Oh, yeah, thank you.